there's so much attention on using CAR T cells for oncology, and without question, there's a major need for addressing those indications. But very intriguingly, what many people don't really know is that original CARs were actually created for HIV as an indication. And at Antion, our origins have been in HIV. We created microRNAs to silence the CCR5 receptor, which is a key receptor for HIV infection of cells. At that point, we were looking to engineer hematopoietic stem cells and to therefore transplant patients with these cells to engineer an HIV-resistant immune system. All of this inspired by the Berlin patient and the subsequent London patient that have been treated. But as we started progressing our technology towards developing engineered T-cell therapies, we realized a great potential to add another layer of benefit to an immune effector cell therapy. And that is, not only can we protect cells from infection by virtue of our CCR5 high efficiency gene silencing, but we could also train these cells to be redirected to target cells that are HIV infected, those that have productive HIV infection. So if cells are infected with HIV, they express the viral proteins on the surface, and therefore they are detectable, and therefore we are able to do that. And we've done something similar to what other groups have done, which is we've engineered the key receptor, which is CD4. HIV would bind to CD4 when it infects cells. But if you reverse the tables and put CD4 onto a CAR, and we created a truncated version of this, you could then create a T cell, which would then bind the HIV envelope protein expressed on infected cells, and therefore elicit a redirected immune response against these cells. And since these cells could also be infected by HIV, and particularly CD8 cells, because CD8 cells normally aren't infected, but because they express the CD4 from the CAR, they would also have the risk of being infected. And this has been shown by us and by other groups. By silencing CCR5, you can protect them from infection. And this is really very intriguing for us to be able to do this. But ultimately, if you want to imagine a feasible treatment, something that's widely accessible, particularly to the people that need it around the world in terms of HIV management, you need something that's broadly accessible and universely adaptable. So what we've done is we've taken the knowledge that we've developed over the years using our multiplex gene silencing approaches and engineered an allogeneic CAR T cell. We're doing it as proof of principle using anti-CD19 CAR T cells, but applying it in the context of HIV is also very interesting, because this then opens the door to a cell therapy product that could be manufactured on a larger scale and distributed globally from a centralized place. So what we've done is we started to develop our technology toward the point where we express a CAR to redirect T cells to find HIV or cells infected with HIV and productive infection, silence the CCR5 to protect them from infection. And by silencing the T cell receptor, we can also make these cells off the shelf. So by silencing the T cell receptor, you prevent alloreactivity of these cells, meaning that I can take cells from a healthy donor such as you or me and give them to an HIV infected patient without there being any form of reaction. So by doing this, of course, you also have the opportunity to expand T cells up to higher fold numbers and to bottle them and freeze them down as a pharma-style type product and to distribute them globally. So if we are to imagine an HIV treatment one day to be distributed globally, this would be the way to do it, in my opinion. CAR T cells have great potential, and there are groups testing the autologous setting, so from the HIV patient themselves, engineering them and giving them back. And I really hope and I believe that there will be promising data that will come from this. We want to benefit from that, but we also want to now add our microRNA gene silencing technology to create a next generation off-the-shelf CAR T cell for HIV.